Okay, this is a quick review of material from Monday's lecture. I didn't like the way I described the lines that we use on PE-pH diagrams, so I'm going to back up a little bit and try to do that in a little bit of a clearer fashion. And notice that we're starting with a list of biologically mediated redox reactions, and that we have a column of PE0, and that is the PE of the standard PE for these reactions. And we also have PEW, which is the PE or PE prime, which is the conditional PE at pH 7 in typical waters. And I want to show you how those two, or how the PEW is calculated. So let's consider the oxygen redox couple, where we have, I'm going to get my pen just a little bit bigger here, where we have oxygen reacting with 4 protons and 4 electrons to give us water as a product. And our PE0 is 20.8. So that means we can write essentially the Nernst equation, where we're writing it in terms of PE instead of E. Let's put the 0 there. And PE equals PE0, 1 over N, 1 over 4, log, products over reactants, and so that's the water concentration squared, the partial pressure of oxygen, because notice oxygen is listed as a gas, and times the proton concentration raised to the 4th power. Now I can break apart that log term, and I'm going to break it apart to remove the proton dependence from the products and reactants. And you notice when I do that, I have this term, and so I still have PE0, I still have a 1 over 4 log, 1 over H plus to the 4th, but now I also have negative log of H2O squared over PO2. So what we've done is we've taken out the proton dependence, and we can lump it together with the PE0, and that is what we're going to define as PE prime or PEW. Okay, well, you notice that PE prime is a function of the H plus concentration, and the log of 1 over H plus is the same thing as the negative pH, and the log of 1 over H plus to the 4th is the same thing as 4 times the pH, and 4 times the pH divided by 4 is pH, and so we see that the PE prime is PE0 minus pH. Well, the PE0 is 20.8, so now we go to our phase diagram, and we go back to, the Porbay diagram. We extend out to 20.8. There it is right there, and it has a slope of 1, so we're going to go over a pH scale of 0 to 14. We're going to go down to a little bit over, we're going to subtract 14 from this, right? And that gets us to right about there, and this is the PE prime for the oxygen-water couple. We call this the oxidizing limit of water because water will actually produce oxygen. If you're more oxidizing, you're going to drive the reaction this direction under oxidizing conditions. And if you drive it in that direction, oxygen's produced. So in any potential more positive, then let's go to pH 7, come up. Any more positive than that potential, we're actually going to produce oxygen. And that's it. And that oxygen would then bubble out of the water. So that sets the upper limit for redox conditions in aquatic systems. Well, notice we have a lower limit as well, and let's go see how we define the lower limit. The lower limit's defined by the reduction of hydrogen ions. 2H plus 2 electrons goes to H2. Its PE0 is 0. We're going to do the exact same thing we did before. There's my PE expression. I've now broken out the proton-specific part. This is pH2, not pH. It's the partial pressure of hydrogen. Sorry for that. I maybe should put a bracket around that, right? And now notice that this term now becomes my Pe prime. And I get 1 over H plus squared, the log of 1 over H plus squared. That's the same thing as the pH, or 2 times the pH. Divide that by half, and we get a slope, again, of negative pH. So it's very common. Not very common. It's just a fact that when your number of electrons and your number of protons are equal, you get a slope of negative pH. And so now we can go back to this slide. We're going to start at 0. Let's put this in a different color. And it's also going to have a slope of 1. And that is the lower limit. Anything more reducing than that line, anything down in here, is actually going to produce hydrogen gas. It's going to bubble out of the system and restore you to this line. So these two lines set the redox boundaries for aquatic systems. And I've done that, right? This box, let's do that with a, let's change that. This blue box is the PE, pH limit for natural systems. All right. Well, that got us the first two reactions. Let's go take a look at some of the others. Nitrate being reduced with by reacting with 2 protons and 2 electrons to form nitrite and H2O. So that is the nitrite-nitrite redox couple. And notice I got 2 protons, 2 electrons. That's not going to be a surprise. We're going to have a line which starts at PE0. PE0 is right here at 14.2. It's going to have a slope of negative pH. And this is the nitrate-nitrite couple. So what does that mean? What it means is that, and I'll switch to blue, if you're above this line, nitrate is the stable species. If you're below this line, nitrite is the stable species. And if you're on the line, nitrate and nitrite have equal concentrations. These are also referred to as preponderance diagrams. In other words, they tell you which species is most preponderant or most common. Above the line, the oxidized species is common. Below the line, the reduced species is common. And we can have multiple lines if we have multiple species. So let's go to the next one. Sulfur sulfate plus 8 protons plus 6 electrons goes to elemental sulfur and 4 water. Right off the bat, you see that the proton electrons are not balanced. So we're going to get a slope that is not 1, but is actually 8/6. So it's a little bit more than 1. It's going to start at this PE0 of 6, so that's where I started. It's got a slope that, again, is 8/6. So it's going away a little faster, and it's also a line on a preponderance diagram. So above that line, SO4 2-, and below the line, elemental sulfur. Said these can get complicated. This is the diagram for iron. Now, iron has multiple species, and the first thing you'll notice is that the oxidation state, and I'm going to make this a little bit smaller and blue, of this species is +3. This iron species, FeOH3, is +3. The oxidation state of iron 3+ is +3, +2, and this species down here is FeOH2 So this is +2. So what's happening in this vertical line is acid base. The iron is undergoing hydrolysis to form the FeOH3 solid species. But what's happening on the more horizontal lines, and they do have a slope to them, this direction is oxidation reduction. So we move from Fe3 plus, the +3 oxidation state, if we move in, I'll do green, this direction, we lose protons or gain hydroxide, so that's an acid-base reaction. But if we move in this direction, we go from iron 3 to iron 2, and so that is a redox reaction. Again, the advantage of these PE-pH diagrams is they show us the preponderant species in each of the boxes. So iron 2 is stable under, the conditions bounded by that box. And iron 3 is stable everywhere in this box, but the FeOH is bounded to the right, and the Fe3 plus is bounded to the left. So now if you make a measurement of the redox potential and you make a measurement of the pH, you now know the dominant chemical species. That is the advantage of these diagrams. If we now go ahead and put all these lines on one diagram, and we circle all the intersecting points, I've done that in red, and let's go ahead and highlight those. We'll do that in a kind of a green. That's right there, and right there, and right there, and right there, and right there. That would say that as you start with oxygen up here, O2, you hit the oxygen water limit right there. And then if you were to keep going, you'd start reducing nitrate to nitrite. And then you've got your iron oxidation kinetics, and then you've got your sulfur oxidation kinetics, and finally you got hydrogen that would start bubbling out of the system. And if you put a vertical line on them, that is the same as the PEW, or the tabulated biologically relevant redox potentials. So here we go. We can take these diagrams, these points on the diagram, we can put them in a table, and we can say if I have electron sources, which is organic material, in this case a sugar, and I have an electron sink, in this case oxygen, it wants to gain an electron, we can start to see the sequence of these reactions. And notice that I have the nitrate-nitrite species, the iron species, the sulfur species, the hydrogen species, and then finally the organic carbon CO2 species at the bottom. The sequence of microbially mediated redox processes. Well, this is sort of a famous figure out of a book by Werner Stumm and James Morgan, and what they're showing in this case is the energy that's derived. Because remember that delta G is equal to negative NFE, and E is equal to PE divided by 0.05916. That's incorrect. Times 0.5916. I'm going to get this right. 0.05916. Okay, so here we go. We're going to highlight the position A there. That's oxygen reduction. We're going to highlight organic matter oxidation. And that is reaction A and reaction L. And so aerobic respiration is A plus L. So we'll go there. And that's worth 29 or 30 kilojoules per mole. So if you're an organism, that is the energetically favorable way to take advantage of your lunch, your sugar. You use oxygen because you get, again, the biggest delta G out of the deal. And let's see. We talked about nitrate reduction. Well, let's change the color to orange. Here's nitrate reduction. We're still going to use organic material as our energy source. That's L and D. And we'll go look at L and D. There it is right there. Okay, so if you run out of oxygen, you can still use nitrate as an electron acceptor, but you're going to give up half your energy. You're going to go from 30 kilojoules per kilocals in this case per mole down to about 19.6. So it looks like you've lost about a third of the energy. And if you run out of nitrate, well, you can always use iron. That's going to be E and L. And when you run out of iron, you can do sulfate. That's going to be G and L. There it is. Now you're down to 5.9. There's G and L right there. Now you're down to 5.9, right? So you've lost 80% of your energy. And if you're really desperate, you can do fermentation, and that's H plus L, and that's methane fermentation. Regular fermentation is F plus L, and that's just going to make, in this case, alcohol. And you notice that regular fermentation is around 6.5, 6.4 kcals per mole. So there's a lot of ways that biology has learned to take advantage of organic material. Oxygen is certainly preferred, and that's how almost all mammals get their energy. But in the environment, there's a whole series of alternative electron acceptors, and you're going to start with the ones that give you the most energy. You're going to use them up, and then you're going to change. And so that's what I'm plotting on this axis. I'm plotting the PE here, and I'm plotting the concentration of organic material, and I'm assuming it's interacting with natural water. And so we're doing a titration, and we're saying if I have a lot of oxygen, or rather a lot of oxygen relative to organic material, my PE is going to be up around 15 or 16. Well, let's go take a look, see if that makes any sense. Sure enough, I'm right up here at 15 or 16. And my redox potential is going to stay there until I run out of oxygen. And as I run out of oxygen, I move down this curve, and then I'm going to start using manganese, and then I'm going to use my nitrogen species, then I'm going to use my iron species, then I'm going to use my sulfur species, and then finally down here, I'm going to start during fermentation. And the horizontal distance you need to travel is really related to the moles of oxygen or manganese or nitrogen or iron that's in the environment. So you're essentially titrating your way along by adding this reduced carbon and letting biology do a chemical reaction. So now we're going to go back and put this in the context of the lake. And this is the diagram I was least happy with on Monday, and so I've redrawn it. Remember that if you take phosphorus, PO4 3 minus, one of those, 106 CO2s, 16 nitrates, and you go this way with sunlight, you can produce organic material. And there is my bug. Now that photosynthetic reaction produces oxygen, and that oxygen is equilibrating with, go away Siri, the atmosphere. And so there's oxygen bubbling out of the system. If you produce excess oxygen and you supersaturate the system, it will escape, because oxygen has a finite solubility. We know that. Well, this bug can fall to the bottom, which we call the biological pump, and it's going to fall to the bottom or through the thermocline into the deep water. And as it falls into the deep water, it now is going to undergo respiration because there's no more sunlight. The plant dies. The bacteria want to eat it. And we now have the release of carbon dioxide, nitrate, and phosphate into the deep water. We also consume oxygen. Well, one of the things that happens as you have anoxic sediments, and under anoxic conditions, you produce iron 2. Iron 2's, again, that iron 3 is the electron acceptor for organic material. Iron 2 is fluxing out of the sediments. And if they leave enough oxygen in the bottom water, that iron 2 gets oxidized to iron 3. That was the homework problem that you handed in yesterday. That iron 3 will precipitate to form iron oxyhydroxides. And notice that is a solid phase. That solid phase will pick up the phosphate. And you now have a mechanism of removing phosphorus from the deep water of the lake. The iron oxides produced when iron 2 is oxidized to iron 3 produces an absorptive solid phase. We're going to come back to that reaction in a little bit. It reacts with phosphate, it precipitates, and it ends up in the bottom, and we call that FEP. All right, so we have, FEP and FEP, and that is a good sink for phosphorus. Well, what happens if you run out of oxygen in the bottom water? Because there's only so much oxygen in the bottom water, and if you run out because you put too much organic material, you titrate the bottom water with lots of organic material. Well, then you don't form any more iron 3 oxyhydroxides. You dissolve your solid phase. When you dissolve your solid phase, your phosphate no longer gets absorbed, but it can be released back into the surface. And when it's released back into the surface, it now promotes additional plant growth. This is what happened on North Pond. The iron oxyhydroxides that build up in the sediments were released when the sediments went completely anoxic. It released enough phosphorus that the phosphorus triggered a massive bloom. Of course, that massive bloom then created a feedback mechanism where we pumped more organic material to the bottom that consumed every last little bit of oxygen, completely anoxic in the bottom 2 meters of North Pond. That dissolved more of the sediments, released more phosphorus, and perpetuated that negative feedback, which caused the bloom. Okay. So how does that all fit back into our pore bay diagram? Well, the green dot is the position of the sediments under anoxic conditions, Right? That's the oxygen-water top limit. Under those conditions, we're clearly in the top box. And in that top box, we have iron 3. Iron 3 binds phosphorus. As we suck the oxygen out of the system and we have enough organic material, we move to the bottom box. And in the bottom box, we're at iron 2. Iron 3 dissolves. Iron 2 does not bind phosphorus. And the Porbet diagram predicts the release of phosphorus in much the same way as our cartoon releases phosphorus. So in one case, we're showing the flux in sort of physical space, and in the other case, we're showing the release of phosphorus in a redox diagram space. So how do we fix the problem? Well, we fix the problem by putting in a redox active species. Putting in a species that's not redox active. This is the stability space for aluminum. Everywhere inside that box you have aluminum 3 that's stable. And aluminum 3 precipitates to form the alum absorptive phase, or alumina, gamma alumina if you want to be specific. So this whole space is essentially AlOH2 plus and all of its acid-base species. That's all in here. And it doesn't care what you do to the oxygen concentration. It continues to bind phosphorus. So if we add enough aluminum, we bind the phosphorus, we break the cycle. And we took advantage of the redox insensitivity of aluminum to break the redox cycle involving phosphorus and iron on East Pond, and we're investigating whether we need to do that on North Pond. Okay, so that's essentially what Abby Sykes has been doing for her senior thesis. She's been looking at the really green North Pond, the fairly clear little North Pond where the sediments did not go anoxic. We've got our redox diagrams here. This is a poster that she presented at the Maine Water Conference. And the bottom line in these two figures is this is the amount of phosphorus that's released from the sediments as a function of aluminum concentration. So you increase the aluminum and you decrease the phosphorus. It turns out that this is the aluminum to iron ratio. And this is, again, P going up this way. And so what's interesting is if you take all the experiments that she ran on all the different lake samples on a bunch of different lakes, and instead of just plotting the amount of aluminum we added, but the aluminum to iron ratio, most of the data follows this really nice curve. And the break right here is 3-to-1. And that means that when we have 3 times the aluminum as iron, we shut down the release. So what's encouraging to us is this makes complete chemical sense. We're breaking the redox cycle by putting in extra aluminum, putting in a redox insensitive species, binding that species through absorption, and holding on to that species even when the system goes anoxic. So that's pretty cool that we can now have a good chemical understanding of why empirically we've been adding aluminum to iron to lakes for now a couple of decades. So what we've really been studying is the biological oxygen demand. Maybe we should call it the biological electron acceptor demand in natural systems. In engineered systems like sewage treatment plants, they actually measure this, and it's the quantity of oxygen utilized by a suitable microorganism in 5 days. So sewage treatment plants take a water sample, water. They make sure it's got the right microorganism that they're interested in, or they use a natural organism. They take a bottle, they measure its oxygen concentration, they cap it, they put it in a incubator for 5 days. They measure the oxygen concentration, and that tells them how much oxygen is being consumed. And they hope it doesn't go to zero, because if it does, it means that those water samples are going to go anoxic in less than 5 days. Why 5 days? Because it was the length of time that rivers would take to flow to the ocean in Great Britain. So you want an oxygen demand that maybe was, less than 50% of the oxygen consumed in 5 days. Any more than that, you had a redox problem. You had your rivers going anoxic or dying as they flowed to the ocean. We can do the exact same thing if we want to just leave the biology out. We can do a chemical oxygen demand. We tend to use dichromate because it reacts quickly with organic material. So if you want to have a quick and dirty technique that doesn't rely on incubation, you can do a redox titration. And so in this case, the chromium is going from, let's calculate its oxidation state. It is 7 oxygen, so that's minus 14. This whole thing's minus 2, so that means we need plus 12. This is going from a plus 7 oxidation state to a plus 3. All right, so the chromium's being reduced. Let's check the oxidation state here. The oxidation state on simple carbohydrate, the carbon is 0, and the oxidation state on carbon is plus 4. And so my carbon is being oxidized and my chromate is being reduced. Again, chemical oxygen demand when we use a chemical species instead of naturally occurring oxygen to drive this organic matter oxidation. Why do you care about that? Because our waste, our sewage, contains oxygen-demanding substances. So what does sewage contain? It contains things like organic material and ammonia and iron and things like sulfite. And all of those things consume oxygen. And here I've given you a bunch of balanced reactions of the reduced species, which is going to get oxidized, and then the oxygen, which is going to get reduced. And where do we do these reactions? We do these reactions in sewage treatment plants. So, this is Colby. All right, we're producing a lot of waste. Our waste goes in pipes. Those pipes flow downhill toward the Kennebec River. They go underneath the Messalonsky Stream, and then they're pumped up and over to the Water Street Sewage Treatment Plant. Primary treatment means they just put a screen in here. It's pretty disgusting, actually. The screen just takes out big pieces of stuff. Wood, pantyhose, clothing, all sorts of disgusting things that get into the sewage treatment. So that's the primary treatment. There's nothing very good about it. It just takes out big chunks. And then it goes into what's called secondary treatment. And our job in secondary treatment is to take out this oxygen demand. Because if we put it right into the river, it would suck all the oxygen out of the river. So here's my simple organic material. It's reacting with oxygen, producing CO2. So now my carbon's leaving the system in the form of CO2. It's producing water, and it also produces biomass. And this is called an activated sludge process. And what makes it activated is you're growing bacteria. So this biomass is actually bacteria that's growing by using the food, which is our human waste. Well, these organisms will take organic nitrogen and turn it into ammonia. It'll take organic phosphorus and turn it into phosphate. these species, the ammonia will actually leave the sewage treatment plant through gas exchange. The phosphate will absorb onto the biomass. And then the biomass comes over this direction and it settles. And so it settles into what we call a secondary sedimentation area. So we have lots of mixing in this area, and we don't have mixing in the sediment area. So the sludge settles out, and then we take some of the sludge and we return it back. Okay, so that keeps the process going, and what really keeps the process going is the added air, because the chemical or biological oxygen demand in this system is huge. So if you left the air out, it would stop. So we keep adding air. They have big bubblers underneath the primary biomass production tank, and it bubbles 24 hours a day. It produces lots and lots of biomass, keeps the process going, and then this ends up going to a truck, which is then taken somewhere for dispersal. And this is one of the big controversies right now, is that the perfluoridated compounds that are a byproduct of paper making have made it into some of the waste treatment facilities in central Maine. And then that sewage sludge gets carried by trucks onto farmers' fields. The PFAS gets spread on the fields because it's such a stable molecule. The sludge contains the PFAS and it builds up in the environment because it doesn't degrade. So as a general rule, sludge is good. It's a good compost material. It's made up of healthy biological cells, but sometimes it includes things that aren't so great, like heavy metals, and in this case, PFAS. And so stay tuned on that one. This is going to be an important environmental concern that I think we're going to have to tackle for many years into the future. Okay, well, that's where I'm going to stop in this narration, and I will talk a little bit about sewage specifically in class on Wednesday, but what I'm really going to talk about is this diagram, and this is the arsenic redox cycle that happens in particular in Bangladesh, and we're going to talk about how that redox cycle has been perturbed by human development, and then we're also going to talk about how we can find solutions to the arsenic problem by using creative applications of redox chemistry and solid absorption of the arsenic to engineered solid phases. So stay tuned. We're going to do that in class tomorrow.